overview of the influenza virus replication strategy, and it begins with the single-stranded RNA genome. And in this particular case, you have a minus strand, remember that, and a preformed replicase. And so the virus is actually bringing with it the replication machinery, some of the replication machinery that is required. In this particular case, the polymerase that allows it to go from a minus strand RNA to a plus strand of RNA. Just remember, we don't have anything like that in our genome or in our cells, and so the virus has to bring that along with it. This plus strand of RNA is basically messenger RNA that can be translated into capsid and envelope proteins. The plus strand of RNA is then replicated using the preformed viral replicase into a minus strand. And then that minus strand is actually genomes. And so those genomes and these capsid and envelope proteins together can make more progeny. So looking at this is just the structure of the 5' cap and of messenger RNA. So remember that mature messenger RNAs have to be capped with this 7-methylguanosine and tailed, so that at the end of this there would be a poly A tail, and that matures the structure. So these viruses have a really interesting capability here, and this is called cap snatching, in which the viruses subvert the host cell's translation machinery to basically turn their minus-stranded genome into what appears to be a plus-stranded RNA. And so in this particular case, the viral replicase is capable of grabbing a 5' cap off of a mature host mRNA. And so in this particular case, it grabs the cap, and then there's the cap, and it uses that essentially to prime the replication of the rest of the minus RNA. And so now this piece of what should be minus RNA has a cap on it, and so now it can behave like a plus strand of messenger RNA and be replicated, translated. Here's a bit of the budding of the influenza virus to the host cell membrane. And so again, this process involves first making RNA genome segments and associating them with nucleocapsid proteins. Ultimately, matrix proteins have to be involved, and then the spike proteins. And so all these things are being made in the cytoplasm of the cell, or translated, I should say, from the viral messenger RNA, and then poked into the membrane. So matrix proteins, the neuraminidase and hemagglutinin spikes and so forth are poked through the membrane. And then ultimately it associates with the viral RNA pieces. And then you start to get what's called a budding event to take place, where the virus literally buds off, takes with it that envelope, as you see here. And then the virus has to pinch off and go out into the world. But it's a little bit trickier than that. And so that's where the virus... that's where the neuraminidase comes in from the virus. And so you can kind of see that these hemagglutinin spikes that you see here could potentially serve as an anchor that would prevent this guy from being released, because remember that this envelope material has sialic acid associated with it. And so if this particular spike right here finds some sialic acid right here, it would prevent this guy from undocking. It's almost like a boat that hasn't, unmoored from a dock. And so something has to cut that rope in order to unmoor the boat from the dock. And what cuts the rope is the neuraminidase. And so the neuraminidase here and here would cut any sialic acid hemagglutinin contact to allow this guy to bud off completely. And so antigenic shift in influenza viruses is what I was alluding to before in terms of the incredible ability of influenza viruses to mutate, as opposed to SARS-CoV-2 and a lot of other kinds of viruses. And this antigenic shift property is why we often see waves in different flu pandemics. So if you have two different kinds of influenza virus 1 and virus 2, infecting the same species of animal, so in this particular case a pig, in case you didn't know what this was, you can get something called reassortment to happen. And so in this particular case, virus 1 and virus 2 infect They dump their segmented genomes into the cytoplasm, but there's a little bit of mixing as a result of these things being thrown together. It's like playing, a game of 5-card draw cards, a poker rather. And so in this particular case, some of the viruses that package their RNA are exactly like their parent strain. And so you see that here with these guys. Not exactly. There's always going to be some mutations because the viral polymerase makes mistakes too. But some of these viruses are basically reassorted viruses. So some pieces from this guy's genome are exchanged with pieces from this guy's genome, and so you get this hybrid. And so there's a genetic reassortment. And this virus, then, this new variant can have extreme diversity so that there's no protection. So showing you that, so in this particular case, even though these people here got the flu, the virus changed so much that they can re-get the flu. And again, we don't see that with SARS-CoV-2. That virus doesn't change as much, and therefore it's unlikely that those peaks will occur in the same way. If we do get peaks with SARS-CoV-2, it'll probably be because we all hid so long and never got exposed to it in the first place. So this is just looking at reassortment from a researcher perspective, and so often what they'll do is they'll take different viruses and co-infect a cell and then look at the progeny. And so here's 3 different kinds of progeny. Here's a recombinant progeny. So you have the L form, the M form, and then this recombinant progeny. And so you can actually use this kind of a gel approach to visualize the recombination. So the L strain has this particular pattern for the different pieces of RNA that occur in the virus. The M strain has this particular, and then you look at the recombinant strain, and instead of having 1, 2, 3, or 1 and 2, this guy actually has a third band in there that represents a hybrid between these two. But all the rest of it lines up pretty well. And so this is just an example. So you can figure out which of the fragments the recombinant strains have, and then you can study the virulence of that. How did that recombination event change the way this virus works? Maybe it makes it less virulent, maybe it makes it more virulent. So other proteins in the envelope are, as I mentioned, the ion channel. So in this particular case, the M2. And I already mentioned how M2 is porous for protons. So it lets protons from the acidified endosome to come in and acidify this internal compartment here, and that acidification allows these spike proteins to change into fusion peptides that allow this membrane to fuse with the host membrane, and so that fusion of membranes allows this guy then to spill its guts into the host. And so you need this M2 ion channel to facilitate acidification of the internal virus core. And so here's another look at of the M2 ion channel, and then here are the main amino acids and how they sort of interact, how they're juxtaposed, how they're laid out so that they can facilitate making this channel. And here's an example of a drug called amantadine, which fits right in here and blocks the channel and prevents this guy from uncoating. So that leads me to this final slide showing you some of the different antiviral drugs. So in this particular case, we're looking at the virus binding, endocytosis, and then the virus comes in, and here's the proton that goes through the M2 ion channel. And so amantadine will block that process by plugging up M2. And obviously it'll prevent membrane fusion and the release of virus RNA fragments. But if you don't have any amantadine, the virus will go through this process and will spit its RNA into the cytoplasm, and the RNA will move through nuclear pores. And then remember, it's a minus strand, so the minus strand will encode a plus strand, and then the plus strand of messenger RNA will be used for additional replication to make more genomes. But it will also be used for proteins. The plus strands will also be used for making proteins by translation. And so Some of the proteins are fully formed M2s that get migrated up here and stuffed into the growing virus structure. So imagine the M2s can get blocked as they're being made by amantadine. So this virus would then get stuffed full of M2s that are already plugged. And if this virus buds out into the world with plugged M2s, it's not going to be a very potent virus. Another drug that's widely used is ribavirin, which interferes with viral RNA polymerase. And so you can hit the virus here as well, and hopefully try to slow down the viral RNA polymerase. And another drug that's more commonly used is oseltamivir, or Tamiflu as it's called. And Tamiflu or oseltamivir is a sialidase inhibitor, so it blocks the neuraminidase activity. So when this virus is trying to bud off into the world, remember I told you before that it's still moored to the dock. And so because the hemagglutinin will find some sialic acid and won't release. And so you have the neuraminidase, which will cut that and allow the release to happen. But imagine if you have an inhibitor of the neuraminidase, like Tamiflu, will keep this virus stuck here in the presence of that flu drug. And as a result, you can slow down the infection process, and then your immune system can come over and beat this guy up while it's tethered to the dock.